In this section, we'll be talking about second sexual reproduction method in bacteria, which is bacterial conjugation. Now, bacterial conjugation was seen by Lederberg and Totem in E. coli. Always remember the name of the scientists, those have worked for that particular sexual reproduction method, and the bacteria they have worked upon. So, if you remember for transformation, I've told you the scientist was Griffith and he worked upon Diplococcus or Streptococcus pneumoniae. Now, Lederberg and Totem have worked on E. coli and gave the concept of conjugation. I've already told you that let's suppose this is a bacteria, and in this bacteria, let's suppose this plasmid is there, and this plasmid is carrying the F factor. If you can look, this plasmid is carrying the F factor. F stands for fertility factor, and that plasmid becomes F plasmid. If somewhere you have the fertility factor, that particular bacteria will be called F plus or male. Male means it will be donating the DNA. So this is also called donor. This particular bacteria does not have F factor. When the plasmid it must be there, but the plasmid does not carrying F factor. And that particular bacteria, when it is not there, when the F factor is not there, when the F plasmid is not there, that particular bacteria will become F minus, or it will become what you call female. Now what happens? Because F factor forms the pilus. This F factor it's forming the pilus. I've told you pilus is required. Pilus you must be knowing about. I've told you about pili. Pili is the plural form. Pilus is the singular form. So pilus is formed, and I've told you that pilus helps in conjugation. So this is what I'm trying to tell you, that how pilus is helping in conjugation. So this pilus, when it is formed in male, it is not formed in female. The pilus will attract this particular bacteria and form this sort of bridge, which is called conjugation tube. The method is conjugation, the tube-like structure between what you call two bacteria which has been formed by the pilus. It's called conjugation tube. Now from the conjugation tube, this particular bacteria will try to send this particular plasmid, let's suppose, to this bacteria. The plasmid is going, if you can look, the plasmid is going, but the plasmid will be going after duplication. So plasmid will duplicate, and after duplication the plasmid is there here. So now you can see this was the donor, this was the male, but the plasmid has already come to this particular bacteria also. So now this also will behave like donor. So there are two donors now. So this is all about conjugation, where the bacteria were combining. In transformation experiment, remember, the bacteria were never combining, they were never joining up. Here the bacteria are combining. There the bacteria, one bacteria was taking up the DNA from the second bacteria, when second bacteria have removed that particular DNA to the media. But here both of the bacteria are combining, and then one DNA is being transferred to the next bacteria, and the next bacteria has started behaving like a new donor. So this is about conjugation, which is also the sexual reproduction strategy in bacteria. In next session, we'll be discussing about the third sexual method, which is transduction.